Hello everybody, welcome back to another lecture video. Today we're going over lecture 22. Doing all these in series, so my voice is slowly starting to go again, as it always does when I do a series of these videos. So rest in peace, my voice. But yeah, let's go ahead and get started. Today we're covering the citric acid cycle, and we're going to dive a little bit into electron transport chain and reduction potentials, basically. So, I already talked to you about how citric acid cycle occurs in the mitochondria, right? Basically, two acetyl in the first round, of any given citric acid cycle, um, you get this acetyl CoA group that comes in, and then eventually those two carbons from acetyl CoA end up leaving. Right, and that process generates a bunch of electron carriers, which we can use in electron transport chain. Rest of this cycle is not really that important. I mean, you can definitely try and memorize this equation, but it's going to be so much easier for you to understand how the actual equation works when you do the actual reaction step by step. So here's the entirety of the reaction, and I would say what I recommend you do is you memorize the name and the structures of every single one of these. Right, that's the first thing I would start with. Right, memorize the name and the structure. That's the first thing I would memorize. Then I would start thinking about some other stuff, but we'll go name and structure first, right? After you memorize, the name and structure, I would then move to the enzymes. But let's go ahead that. So let's cover all of these, right? So basically, right, we've got citrate, right? Citrate synthase is very simple, right? It synthesizes citrate, right? Aconitase is a bit of a weird one. You can just think of it as an isomerization. See how this OH is moving from that carbon to this carbon, right? Um, we have a dehydrogenase, right? And I want you to think about what dehydrogenases do. Dehydrogenases do redox reactions. So I can expect to see either NAD plus or FAD taking part in this reaction, or NADH or something like that, right? I expect to see a electron carrier here, or like an electron, like an electron carrier of some kind here, right? This is alpha ketoglutarate. The other thing you should notice is that this structure is very similar to glutamate. Glutamate is just this structure, except this O here is replaced with an NH3 or an H2, right? That's glutamate. So it's literally just the amino acid, but with an amine switched out for a carbonyl group, right? That's the other thing you should notice. Right? So after alpha ketoglutarate, we have alpha ketoglutarate dehydrogenase, right? Which does a dehydrogenation reaction. So I'm expecting to see some sort of like NAD plus NADH, FAD, FADH2 type thing, right? Succinyl-CoA synthetase, right, with succinyl-CoA. So succinyl-CoA. So notice how this S-CoA came into play here. It wasn't here before, so it must have been added in this step, right? That's why I'm saying if you memorize the structures, then you can kind of figure out what goes in and what comes out, right? Succinyl-CoA, right? You have succinyl-CoA synthetase, right? Synthetases, right, generally speaking, take GTP or ATP to form something. But in this case, the enzyme was actually initially discovered in going in this direction, so that's why they call it a synthetase. But it actually ends up producing GTP slash ATP, which we'll talk about later. But it's called a synthetase, right? Because it was discovered going in this direction, right? And when it's going in this direction, it has to actually use GTP to make succinyl-CoA, but in the other direction, it actually uses succinyl-CoA to make GTP and succinate, right? Once we're at succinate, right, we do succinate dehydrogenase. Again, a dehydrogenase, I'm expecting an FAD, FADH2, NAD, NAD plus, or whatever, NADH, right? Something like that, right? Then we go to fumarate, right? And from fumarate, we're using fumarase, and we end up with this OH on one of the carbons. So the only way I know of adding an OH in biological conditions is through water, right? I can't just add hydroxide. Hydroxide is not really within your body that much, right? So I'm expecting to see some water going into this reaction here. So maybe a water is going in here, right? So, and then if we look at this, right, I've got another dehydrogenase. So same story. I expect to see NADH, NAD plus, FAD, FADH2, stuff like that, right? And the other one I want to make note of is the structure of oxaloacetate. So we talked about alpha ketoglutarate, right? And how this turns into glutamate, right? If you replace this O with an NH2 on oxaloacetate, it turns into aspartate, right? So I would try drawing these two out, right, on your own, and just seeing how that's true, right? Seeing how these two amino acids are actually just like can be directly fed into citric acid cycle through that kind of just changing of a group. You don't have to know about the enzyme that does that. You'll learn about it in 441, but if you're curious, it's called transaminase. Um, and it basically exchanges a carbonyl group for an NH, an amine group, right? And the other one that has a similar property to this is actually pyruvate. So if you remember, pyruvate actually inhibited a lot of the, or sorry, alanine, sorry. Alanine, right, is similar, right, to pyruvate. I feel like I've drawn this wrong. Yeah, I have. So alanine, right, is actually the same thing, right? Alanine, if you just switch out this O here for an NH2 group, NH3 plus, right? That's alanine, right? So if you remember from glycolysis, alanine actually inhibited some of the enzymes like pyruvate kinase, if I'm not mistaken. That is why, right? Because alanine is just a side product or a transamination reaction of pyruvate. So if we now move on to citric acid cycle, right? We've got all of these, right? I don't know why I said move on to it. I was looking at something else. Sorry. Okay, so now we're back here, right? So now we're talking about labeling carbons, right? And labeling carbons is, in my opinion, one of the more difficult aspects of citric acid cycle, and being able to trace carbons through the throughout the process, basically, right? So the main idea they're trying to get across here is that the first two carbons of acetyl-CoA do not come out as CO2 in the first cycle. They come out in the second cycle. So let's imagine, right, that we start with acetyl-CoA, right? And let's just say that I'm labeling this carbon and this carbon. I know they've circled stuff, but I don't really like how they circled it, right? But let's say I'm labeling these two carbons, right? Let me actually use a different color for this one here. Orange, does that work, right? Yeah, so I've got orange and red, right? It's kind of hard to see, right? But orange and red, right? So I've got an orange and a red carbon. When I attach, right, the way this is going to work is we're basically going to have like an enolate reaction where this carbon here is going to end up attacking here, right? And it's going to make this OH group here. This is going to go out to the side right here. And we're going to be left with this. So now the C, that orange C is here. And the red one is up here. Right? So now if we keep going down, right, nothing really changes. The red one is here. The orange one is here. Same thing here. The orange one is here. The red one is here. So nothing really changes, right, for these first couple. We keep going down. The red one is here. Still nothing changing. Oh, that. Okay. Now we're getting to a bit of a weird territory where once the loss of this acetyl-CoA goes away with that decarboxylation, or not decarboxylation, loss of that acetyl-CoA is, I have no idea which side this COO minus and this CH2 are on, right? Because it's symmetrical, right? And because it's symmetrical, I can't tell if this group here has been flipped to go on this side or if it's still here. So it could be either one, right? So it could be the red ones here, it could be the red ones here, it could be the orange one is here, the orange one is here, right? And you basically have that going up as well, right? The orange one could be this carbon, the red one could be this carbon and this carbon. Same thing up here. This could be the red, this could be the red, this could be the orange, and this could be the orange, right? And the same thing up here, orange, orange, red, red, right? So I have no idea, right? But that's the first cycle done now, right? Those are now the possible locations of the carbon from this acetyl-CoA. Now, if we move into the second cycle, I just said that, I don't know based off of this where they are. So let's say I've got my red here, and I've got my red up here. I've got my orange, right? And so now when my next acetyl-CoA comes in, right, I'm going to mark this in green, right? These two carbons, right? Actually, I'll mark that one a different color as well. This green, this purple, right? Now, right, I'm looking at this again. So this C, right, this COO, right, is here. This this carbonyl here gets attacked, right? So it's here, or it could be here actually, right? So it could be that one too, right? Because it could be this carbon right here that got attacked, or that it could be that the orange is actually this carbon, right? And then we've got the purple that attacked here with the green on top, right? So now you can basically go through this cycle again, right? And if you look at isocitrate dehydrogenase and alpha ketoglutarate dehydrogenases both release CO2 during their reactions, right? And what ends up happening is you first release this CO2 and then you release this CO2. And so that's when you actually lose the carbons from the previous acetyl-CoA. So you don't actually lose the carbons from the previous acetyl-CoA until you get to the second cycle, and basically like 3/8 of the way through the second cycle, right? That's when you actually lose the first CO2. Actually, also when you lose the second one. I don't know why they haven't said, they only said one here. Both of them should come out. So this one comes out and this one comes out. It's possible, right, that some of them don't come out depending on like the location, right, of the carbon, but they have to specify which carbon they're labeling. And usually they only ask you to track through one cycle, because if they ask you to track through multiple 2 cycles, then it gets very messy. So they'll have to specify when they're tracking through 2 cycles, like what are they tracking, and then where does it end up in the second cycle, stuff like that. So you need a little bit more information. So the net result of citric acid cycle is this. There's CO2 released in, I didn't really mention that before, but there's CO2 released in these 2 steps here, right? So notice how I talked about where I expect NADH and FADH to show up, right? So the dehydrogenases all have like the usage of this. So NAD plus there, NAD plus here. The synthetase has this here, another dehydrogenase, and another dehydrogenase, right? And then there's water here as well. Okay. But notice how the dehydrogenases all use NAD and FAD, right? The main thing I want to note is when they use this. So for all of the decarboxylations or the oxidations of like a carbonyl to oxidations of an OH to a carbonyl, it's using NAD. But if it's the removal of 2 hydrogens here, right? So notice from succinate, we're removing one of these hydrogens from each bond. We use FAD because FAD grabs 2 H's. That's a very simplistic way of thinking about how it works, but you can think of it like that. Basically, you can think of it as the FAD takes 2 H's, and because it takes 2 H's, it forms a double bond. The other ones all use NAD+, and they also release CO2, except for malic dehydrogenase. This does not release CO2. So, this is just the what the equation is, right? You have 3 NAD pluses being used, so isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, and malate dehydrogenase. You have succinate dehydrogenase for this, succinyl-CoA synthetase for GDP and PI, and then you've got fumarase for the water. That's where all these reactions end up using these substrates. And of course, acetyl-CoA, right? So what you should note is, right, the number of carbons that acetyl-CoA starts with, right, is 2. We got 1 carbon and 2 carbon hidden behind here. And the amount that is released is also 2. So it's a cycle. We're actually not gaining or losing anything net. The net gain in carbons is 0, and the net loss in carbons is 0. So we actually don't lose anything. We're putting stuff in, and that stuff that is being put in eventually all comes out, right? So we start with 4 carbons with oxaloacetate, and we end with 4 carbons again by releasing 2 CO2s. And here we talk about this again, right? In one turn of the CAC, 3 NAD pluses, and it should say FAD, not FADH. And 1 FAD are reduced to 3 NADHs and 1 FADH2, right? We already talked about this, these reactions here, and we already talked about why this one's FAD and how you can think about that. And here's just a nice little diagram, or like a accounting thing of how much stuff is released. If you look at this is pyruvate dehydrogenase right here, so PDH, right? This is ETC. So if we look at this, right? Pyruvate, so PDH does not produce ATP. It produces 1 NADH from E3, right? And it does not produce NAFADH2. Remember that the FADH2 in pyruvate dehydrogenase is actually just recycled, right? It's not being released into the mitochondrial matrix where it can be used as a carrier. It's just being recycled in that enzyme over and over again. So, and then for citric acid cycle, we've got 1 ATP, 3 NADHs, and 1 FADH2. We've got all of these added up together. 1, 4, and 1. And so you can imagine that if we have 1 mole of glucose, right? 1 mole of glucose is 2 pyruvates. So you'd go through this cycle twice. So you just double everything. So 2 ATPs, 2 NADHs, and 2 FADH2s. But if we look at glycolysis, right, would do per mole of glucose, 1 mole of glucose, you're going to put 2 ATP in, you're going to get 4 out. So, and then if we look at this, we've got the combination of these two, right? We were combining glycolysis with citric acid cycle here. It's kind of weirdly drawn, but, and then of course glycolysis occurs in the cytoplasm, and citric acid cycle is in the matrix. You can see how, the combination of these two things actually produces a decent amount of energy and electron carriers for our electron transport chain. Yeah, so citric acid cycle is we got there's different kinds of process, and we'll talk about it, but so we kind of draw it like this as like a very circular thing, right? But it's actually more like this, where things feed in and come out and all that jazz. Remember how I talked about how you can actually put a glutamate in here, and you can actually also put oxaloacetate or aspartate in here. So like there's all sorts of things feeding into this, right? There's all sorts of other amino acids that can go into malate and all sorts of weird things. So just remember, I'd say the main ones to remember are alpha-ketoglutarate and oxaloacetate. Those are like two of the main things I would remember, is that they are actually just amino acids with their amine removed.
And here's some other examples of amino acids that can go in and come out as to do gluconeogenesis and stuff like that. So you got cataplerotic and anapleurotic processes. So we get anapleurotic builds up intermediates. So aspartate, phenylalanine, tyrosine. These are anapleurotic because they build up fumarate. They make fumarate. These are isoleucine. These things are all anapleurotic because they make succinyl CoA. Catapleuric things are ones that use up the intermediate. So glucose uses malate to make it. So it's catapleuric because using up the intermediates, right? Amino acids, a lot of amino acids like glutamate for this, and oxaloacetate and God's sake, for glutamate for this one, and aspartate for this one. Right? Those are catapleuric because they use up. They're actually both catapleuric and anapleurotic. They actually go both ways. But stuff like that, and then citrate, things like citrate are used for cholesterol, right? These are all catapleuric. You get the idea. If it uses up the CAC intermediates, citric acid cycle intermediates, then it's catapleuric, and if it creates them, it's anapleurotic. And now we can take a look a little bit at energetics and regulation. So if you remember from glycolysis, the big negative steps were steps 1, 3, and 10, and those are very tightly regulated. It's no different here. So the ones that are very negative here are steps 1, 3, and 4, and which correspond to citrate synthase, isocitrate, the dehydrogenase enzymes, and this makes sense. The dehydrogenase enzymes being very negative actually makes a lot of sense because they release CO2. Malate dehydrogenase is a bit of a weird case, but I don't think you have to know it too well. I think it might be talked about a little bit down there, but it's not too big of a deal. So these for sure, the 1, 3, and 4 for sure are the things that I would say are the ones you need to care about as the regulatory steps. And if you're ever asked like why is it one of the regulatory steps, you can just say very negative delta G due to release of CO2 and creation of NADH. And that's one way of talking about it. I want you can also think about like why is it that if CO2 release is negative, why is this so positive? Right? And why is it 0 here? And you have to think about that's probably due to this, right? So we probably have a really massive ratio of reactants to products, right? There's a lot of reactants and not that many products, right? So that would make this whole thing negative. This plus 29 would be canceled out and it'd be about 0. Regulation of the citric acid cycle, you kind of just got to memorize this. Some of them you can like kind of logic your way through. But remember, like I said, steps 1, 3, and 4 are the ones that are regulated, so we can just think of it like that. So if you look at this, what activates citric acid cycle? Anything that says that there's low energy. So ADP. ADP here for isocitrate dehydrogenase. Calcium is what calcium signifies muscle contraction, right? So calcium would also be an example of something that would cause activation. Things that would inactivate these enzymes are things that suggest high levels of energy, right? High ATP levels. So ATP inhibits it, right? Succinyl CoA. Succinyl CoA is a product over here, right? Succinyl CoA is over here, right? That's like a feedback inhibition. Oh wait, not over here. Sorry, it's literally right there, and I can't see it. Succinyl CoA, right? Literally goes back and inhibits feedback inhibition, right? And then NADH, right? These are things that suggest that their energy is high. Citrate for citrate synthase, right? This is another feedback inhibition, right? And then again, succinyl CoA and ADH. So you get the idea. These are things you don't have to memorize these. I would say. I think it's just important to know the general trends because some of these are like really hyper specific. But just know that if it's an energy suggesting molecule, like it suggests that there's high energy, it's gonna inhibit. And if it suggests that there's lack of energy, then it's going to activate these enzymes. So, yeah, we've got the different phases of respiration and all that. I mean, we kind of already talked about these. I'm not going to go over these in depth here. So cellular respiration occurs in 3 major steps, right? We've already gone over acetyl-CoA production with PDH. We've gone over oxidation with CAC. All we have to do now is talk about electron transport chain. So basically, we've generated all these NADHs and FADH2s, right? And these are important if you remember from Bio 200 or any bio class you've taken, really, and even 220 actually to an extent. These FADH, these electron carriers, right, are used to actually pump a proton gradient to power ATP synthase. And this is where the reduction, or not reduction, the introduction of reduction potentials comes into play. So we know that, the oxidized form of something has no electrons and the reduced form has electrons, right? So they are a redox pair, right? You cannot, a redox reaction has to have one thing oxidized and one thing reduced. This is a redox reaction because, well, in this case, it's more, well, I guess it's not really a redox reaction. It's more of, this is the reaction that is required for a reduction to occur with hydrogen, right? That's the way of thinking about it. And the more positive the reduction potential is, so E naught, right? The greater the tendency is to gain electrons. So you can imagine that a more positive E is proportional to a more negative delta G, right? That's not how you draw a G, right? More proportional, it's proportional to a negative delta G because it suggests spontaneity. So we've got, standard reduction potential. I won't bore you with this. I don't think this is really that important. But basically, it's like the standard conditions for the delta G naught prime, where we do everything at 1 molar, 25 degrees Celsius, and we're comparing it to a reference of hydrogen, right? Which is arbitrarily set to 0, right? That's our 0. And then, of course, pH 7. And then the only thing that's different is the amount of H+. So if you do all that for all these different compounds, you get these values. So what this means is oxygen loves being reduced. FAD also likes being reduced. Fumarate also likes being reduced. Oxaloacetate, not so much. NADH, not so much. Isocitrate, not so much. Right? So these guys like to act as donors, basically. Or they like to accept electrons in this case, because they're actually doing reduction potentials. Right? So these guys like to accept electrons, and the rest of them don't. Down here, at least, they don't. These three do. And you can do the same thing with delta G's that you do with these guys. You can add them together using Hess's law, right? So if we're given this reaction and we have to solve it using this half reaction, all we have to do is write it to cancel. So we know that alpha ketoglutarate is on the right side. So we're gonna have to flip this guy, right? Flip this equation. So it's gonna actually end up flipping this to be plus, right? And then we also know that if we flip that, everything else should be good, actually, not mistaken. So this actually can stay as that. So I believe it should be around 0.06 volts. Okay. So what powers this is actually, funny enough, is going from isocitrate to alpha ketoglutarate. That is very stable, right? Very favorable, and that powers the reduction of NAD plus to NADH. Pretty sure I did that right. So you can think which one is the electron acceptor, which one is the donor. If you want to think about it in a better way, right? The better way of thinking about it is probably actually doing it like this. This is probably how they want you to do on the exam, most likely, is which one is the acceptor, which one's the donor. So which one has the more positive reduction potential? In this case, this has the more positive reduction potential, right? So this is going to be the one that's getting reduced, right? And this is going to be the one that's oxidized, right? So because we do that, we do cathode, so it's negative 0.32 minus negative 0.38, which is 0.06 positive, right? So I actually think you should probably use that method more, because if you try and do the flipping, it can get a little bit complicated with how to explain it, but the principle is the same, right? think I actually might have messed up my explanation for that. But yeah. Hopefully that second version makes a little bit more sense. That's what they're usually looking for when you do these kinds of problems. And then we have the Nernst equation. Well, because you don't have what's it called, a calculator on the exam, you can generally just do a little bit of simplifying, where you can simplify RT, this 2.3 RT to just 5, right? And then you can simplify F from 96 to 100. And all you're really doing is you just have to multiply it by n. So you like 5 divided by n times something, times 100, basically. And then you can do the math from there. And that is written here for you. And so if you do, the whole relation of delta G and E, you get this. And that makes sense because we were just talking about how E is going to be proportional to negative delta G, right? So this makes sense. Do you have to know how these equations are derived? No. You just have to realize how this works. So essentially what's going on here, right, is that if delta G is negative, the E is positive, and the reaction is exergonic. If delta G is positive, then E is negative, and the reaction is endergonic, right? So it's actually inverted. So if G is positive, E is negative, and if G is negative, E is positive. And I've actually already alluded to this before, but the electron transport chain occurs in the inner membrane of the mitochondria. So there's two membranes, right? The inner one is where the electron transport chain occurs. And here is the electron transport chain. So the main things you should know about the electron transport chain, here is the outer membrane, and here is the inner membrane. The main things you should know is that complex 1, complex 3, doesn't really show it, and complex 4, they all take NADH. So basically this NADH starts at complex 1, and it goes through all of these complexes. On the other hand, FADH2 only starts in a complex 2. I should specify that NADH from complex 1 does not go through complex 2. But NADH starts in complex 1, goes to complex 3, goes to complex 4, and then this redox happens where it's oxygen is released to water, which is why we need the O2 in the first place, right? So this is why we need O2, and this is why we need to breathe, because we need the O2 for the electron transport chain. So the NADH goes through complex 1, 3, and 4. The FADH2 goes through complex 2, 3, and 4. And because it only goes through complex 2, 3, and 4, it actually misses out on a lot of protons. But that's not in this lecture, so we'll go ahead and probably talk about that a little bit later. I will kind of briefly discuss this as every time, right, an NADH goes through, or like anytime an electron goes through these complexes, right, a proton is pumped from the inner area of the matrix to the intermembrane space, and that goes against its concentration gradient. So we're actually creating a concentration gradient across this membrane, like that, going this way. And notice how if you start with FADH2, you're actually skipping this proton. You're actually not even doing this. So for FADH2, you're only generating these two protons going up, while if you do NADH, you're actually generating all three of them. So that's why NADH actually ends up producing a bit more energy. And I can't remember the exact number. I think it's 2.5 ATP per NADH and 1.5 ATP per FADH2. But yeah, so once those protons are pushed to this side over here, they fall through ATP synthase, and ATP synthase, it was able to turn ADP and PI into ATP. So hopefully that makes sense. It's a bit of a broad overview of how this works. But yeah, hopefully that makes sense. If you have any questions, feel free to stop by the IC and ask me. I appreciate you guys for watching, and hopefully this was at least somewhat helpful. Have a good one, and bye-bye.